Measles is one of the most contagious viruses known and can spread if even a small percentage of the population is unvaccinated. The virus causes a rash, high fever, upper respiratory symptoms, and profound though transient immune suppression. It can also cause serious and sometimes fatal complications, especially in young children, pregnant people, and people who are immunocompromised. Global cases of measles have increased by 30-fold in the last year, and the disease is the biggest vaccine-preventable cause of childhood deaths. The current measles vaccine, which contains live, weakened virus, is safe and highly effective. But because people who are immunocompromised cannot be vaccinated with a live virus, they are vulnerable to measles unless at least 95% of the population is fully vaccinated. Currently, less than 92% of adolescents in the United States are fully vaccinated. With a growing number of immunocompromised people who cannot be vaccinated with a live virus, measles has more opportunities to spread. A new study highlights an innovative approach to measles prevention and treatment, focusing on non-live virus vaccines and targeting viral proteins to safeguard immunocompromised populations and tackle measles complications more effectively. Researchers from Columbia University and the Jolla Institute for Immunology have developed a new approach to combat measles, focusing on creating vaccines and drugs that address the virus's increased global spread. By targeting the measles virus fusion protein, they aim to create safer vaccines for immunocompromised individuals and enhance treatment for measles-related complications like encephalitis. Scientists have been working to develop a new measles vaccine that does not rely on live virus and can be used to protect immunocompromised people. The current vaccine, which contains an inactivated or weakened form of the measles virus, mainly generates antibodies against hemagglutinin, a protein that helps viruses attach to cells. Researchers are trying a different approach, isolating specific components or subunits of the virus to generate antibodies towards other important proteins. Since subunit vaccines do not contain the whole virus, they are safer for people with weakened immune systems. The component the researchers targeted was the fusion protein, which helps the virus enter the cell and cause infection. They found that antibodies targeted towards the fusion protein successfully prevented the virus from completing its merger with the cell membrane. At the same time, scientists had been looking for ways to treat measles encephalitis, an often fatal complication in which the infection spreads to the brain. They found that mutant forms of the measles virus found in the brains of people with measles encephalitis contain modified fusion proteins. When the researchers investigated this variant of the fusion protein with cryo-electron microscopy, they obtained a frozen snapshot of the action at each step, giving them critical insight into how the antibody they created interacts with the virus. Even after the antibody latched onto the protein was able to transform itself into an intermediate state, initiating the fusion of the virus and cell membrane. But the antibody prevented the fusion protein from completing this process, essentially blocking infection. Researchers are now in the process of testing the efficacy and safety of a novel set of stabilized measles fusion proteins as a subunit vaccine for immunocompromised individuals and for vaccinated individuals with waning immunity. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.